So here I'm going to show you the third part of problem 14.3. So this is going to be for Russell iGenetics 3rd edition problem 14.3-3. In the first part we determined that this problem is a cross of a female who is heterozygous at 5 loci, A, B, C, D, and E. Sorry, that E would be wild type, not that. Against a male, males and Drosophila do not undergo crossing over, so he should be homozygous recessive. And his genotype is this. This is not a proper representation of his genotype because the order and linkage is not shown. It's shown alphabetically, and that's a clue that we don't know. So in the book this is given. I typically would not give you that information because it's clear from the problem. And in the first two parts we determined that there's two linkage and that the actual order, the actual map is B, D, A, C, and E on a different linkage group, such that the actual genotype of the parent of interest can be represented where the order, physical order of the loci is shown, the linkage is shown, and the repulsion versus coupling, the cis-trans of this, and that's sometimes quite important. So this then is the genotype of the parent of interest. And now in the third part I would like to show you how to determine relative map distances. So remember, because we have two linkage groups, we have reciprocal fours. We have two reciprocal pairs, and these then represent parental crossing over between B and the remainder. So these represent single crossing over between B and D. This one was B and D were separated from A and C, so this represents single crossing over between locus D and locus A. And the third group here represents crossing over then, what was it on the end, C and the others, so between C and A. So if we had a more extensive problem where instead of the parental one time, and one time crossing over, we had the classes that are so rare they don't show up here, the 3 2 times and the 1 triple crossing over, if we use the formula in the book to calculate precise map distances, we would include the information of these. Here they don't exist, so they don't contribute, and in any case the method I will show you doesn't use them. I will show you a way to calculate approximate map distance, and by approximate I mean we neglect these anyway, and we only use the single crossing over. This is a single crossing over recombinant class, single crossing over recombinant class, single crossing over. What do we see here? We see that 12, 13, that's 25, so that's 30, so this is 55. There are 55 out of 1,000 progeny that are have the phenotype, the result of crossing over in the mother between B and D. So that's 5.5 map units. So if I make a little map, I would say between B and D, for example, there is 5.5 map units. And between D and A, I will count here, I'll say 27, 35, 43, 43 out of 1,000. That's 4.3% or 4.3 map units. And here lastly, between C and A, we see 14, 20, 27 out of 1,000 or 2.7 map units. So here we have a map. So we have different parts of the problem. We have the correct notation of the parent being test cross. In this case, it's probably we can infer that it's the female. All the information is there, and we can determine the approximate distance and map unit between these loci. Notice we're not interested in the distance between E and the other loci because we don't have any information. So in a separate linkage group, presumably it's in a separate chromosome. The use is synthetic, this one's on a separate chromosome, or it could be on the same chromosome but very far away. That doesn't concern us. So I've shown you here how to extract the genotype of the parent, how to figure out the linkage and order, and how to write it properly in notation, and how to determine approximate map distances.